Today's speaker is Micha Drukker. He's a professor of stem cells, development biology, and technology for innovative drug research. And I also know that Micha joined our faculty earlier this year, or at least around December, January. So Micha, thank you so much for joining us and welcome to the faculty. And also thank you so much for sharing some of your work with us. I think it's going to be a very interesting seminar. I saw some of your slides and I'm really looking forward. So Micha, once again, welcome to our seminar and also to our faculty. And I think the microphone is yours.
Thank you so much. Can you hear me well now?
We can.
Excellent, fantastic. Thank you so much. Welcome to my short seminar about making drugs from stem cells. My name is Micha, actually not Micha, Micha Drukker. And I am from the Leiden Academic Center for Drug Research, the LACDR at Leiden University. I need to move to the next slide. See that it works. Yes. So, in case you were wondering, my last name is indeed Dutch, but I grew up in a different country. I grew up in Israel. My father is from the Netherlands, so I definitely need to improve my Dutch in order to have a conversation with you in Dutch. But I'm working on it. Now, I did my PhD studies at Hebrew University in the topic of genetics, and then postdoctoral studies at Stanford University in regenerative medicine. And about 8 years ago, I became a group leader and unit leader at the Helmut Center in Munich, where I studied stem cell biology and cellular reprogramming. And I will explain what that means in the coming minutes. Since the beginning, the beginning of the academic year, I'm a full professor at the LACDR. And you can see on the right-hand side the photo of my very blonde family. And my kids now are learning the fourth language. So they are almost native, I would say, in English, German, and in Hebrew, and now Dutch. So my interests are and always been in mechanisms of stem cells, basically how stem cells create our body, how they maintain our tissues throughout life, how they age, how do things go wrong, for example, in cancer. I'm also very much interested in stem cell technologies, how to use stem cells in order, for example, to understand disease, as well as in stem cell therapies, how to use the cells themselves as a therapeutic entity. And on that note, I would like to explain my goal here at Leiden University. I would like to establish a program around stem cells as drugs. And starting with the statement, I would like to explain that actually drugs that are made of living cells, without a doubt, will revolutionize medicine because the need is so clear. Replacement of damaged tissues. For example, we can see here for the physicist, another scientist, Stephen Hawking. There are diverse diseases, the infinite amount of diseases almost where people are affected, for example, in diabetes and Parkinson's disease, and chronic kidney disease, blindness, and so on and so forth, where replacement of cells can lead to cures. Now, to put this in perspective, most of the pharmaceutical logic and the science has been created around small molecules consisting of few dozens of atoms. But now we're talking about a drug that is entirely different, actually consisting of masses of molecules, and genome, RNA molecules, lipids, and so on and so forth, something much grander. So this requires, in my opinion, an establishment of completely new research areas in the drug development sector. For example, differentiation protocols is the way that we use in order to manufacture the drugs, the cells themselves, from pluripotent stem cells. And this is a new topic, of course, in drug development, because in the past we used to simply manufacture, synthesize the drugs. We need to think about aspects like cell type identity. So what are the drugs, how they are identified? What is the cell type that we are using? There are new questions essentially with regard to the stability of these drugs, with regard to their genome. Now we are using a drug that has a genome. It can go about numerous functions. So we need to take that into account and focus on that as well, because we don't want mutations to accumulate over time, for example. We need to think about long-term activity. So classical drugs are active for hours, or sometimes days, sometimes weeks. But here we're talking about drugs that will be basically having their effect for years and even decades. So that also brings a completely new dimension of the drug program. Tissue engineering is, of course, very important for obvious reasons, because we need to put the cells in a certain context, not just inject them randomly. And as well as a new topic is the stem cell graft immunity. So the cells, if transplanted with me and Pedro, for example, are very likely to be rejected because of immune rejection processes. So we need to think about that and how to overcome these hardships. And finally, I would say that we need to think about very practical things like economics and the business development around these drugs, because they are a new case. They are currently very expensive to make. So we need to find ways in order to make social impact with these drugs. Now, I would like to explain a bit about my approach in science. In essence, my approach has always been in the axis of development, two topics, fundamental and applied. So I study stem cells because it's extremely interesting to understand basically our own natural history, how we come to life. So stem cells are unique in the sense that they can self-renew, they can recreate themselves, and they can also at the same time differentiate to specific cell types. And stem cells exist, and that's probably a topic for another lecture. They exist in the early embryo, in the developing embryo later, and also in our tissues. Some tissues contain stem cells while others don't. And they are, of course, implicated in cancer. They also age over time, leading to our aging. So there are many beautiful questions around stem cells and their function that one can study, I mean, in a very basic fashion in order to understand basically our life. But also stem cells can be directed to, simply put it, replacement parts. And that's also an axis of research that I do very actively, because I would like to take the fundamental research and combine it with applied purposes in order to basically enable therapies. So what my lab works on is a specific type of pluripotent stem cells, the earliest stem cell, if you will, the induced pluripotent stem cells. And we produce them by a process called cellular reprogramming. And for you to think about this, cellular reprogramming is, in essence, is like a time machine, that we can take some fibroblasts or from the skin, even some cells from the blood, or from urine, from leftovers, things that come out from our body, and actually reprogram them. And what you can see here in the background is actually a colony of induced pluripotent stem cells, and each small dot is a cell that have been grown into this colony by cell replication, simply. So we call this a clone or a colony. Now the part of the time machine is in place again. We can, back to the future, we can differentiate the cells again into for example, motor neurons, into lung cells, heart cells, pancreas cells, and we are getting better and better in doing that using something that we call differentiation protocols. So you can think about this as basically factors that we apply that mimic the way that cells develop during organogenesis in embryonic development. So differentiation protocols, of course, very different between different cell types because they represent different cell types of the body. Now, this enables us to do, I think, really great things. For example, to study our own developmental history. How do we form from this early Big Bang and develop into what we are? It allows us now, by differentiating cells, to understand disease, for example, congenital disease, like we do in my lab. It allows us to study drugs, perhaps, to correct disease as we see it in the plate, using those cells that are embryonic-like cells. They're not embryonic cells, but they are like embryonic cells. And we can also think about aspects of toxicity, how to find out drugs that have less toxic effects, and also basically to look at toxicity in environmental factors. That's also very important. Without exposing animals to these compounds, we can simply do it in the plate. And as I said, another very important topic is the cell therapy, basically to create cells, transplant them in people in order to cure certain diseases. And together with LUMC, for example, here in Leiden, we work together on cures for diabetes by differentiating beta cells from induced pluripotent stem cells. Another aspect that I will mention in the end, I think is a really fascinating prospect to use iPS cells, and I call this approach iPS zoo, but using induced pluripotent stem cells, these early cells actually from different animals, different mammals. And that creates, I think, many interesting possibilities in order to understand the dimension of development, time of development, and also maybe also to make sure that certain species that are going into the extinction phase are preserved, at least in our freezers for now. I will mention this in the last slide. So I would like to show you some questions that we are dealing with that I think that are very fascinating. And I think one of the basic one, I mean, for physicists, for chemists, for biologists, for anyone, is to ask what is the basis for stem cell regulation. In the past, we used to address these questions by using, for example, mouse embryos. You can see here a mouse embryo, say day 9 of development. But this approach has many limitations. It doesn't allow us to isolate large regions or many cells in the mouse embryo because it's extremely small. We need to sacrifice many animals to do so. And of course, mice are mice. They're different from humans. And this approach, therefore, has limitations. Now, using stem cells, and you can see here nuclei of stem cells showing here that are differentiating to the right-hand side. I will explain what these funny dots are. We can understand how genes work in the regulation of our coming to life, of our tissues. We can understand what are the aspects of epigenetics. Epigenetics is the science where we ask how genes embedded in the nucleus, in the DNA, are being activated by the landscape and the folding and the enclosure of all the genes in the genome. We're asking how the living genome is important in order to express certain genes or to suppress others. We can study how metabolism is affecting development. We can ask questions about gender. We can ask questions about the genetic background. We can ask questions about aging, how they affect stem cells, and so on and so forth. So the power of stem cells I think is extremely great. Is great compared to the previously, where we were limited more to mouse embryonic development. And I've placed this example of our own studies about stem cell regulation, an example that I think will be attractive to chemists and to physicists, because the process here that is implicated in the differentiation process is actually phase separation. You can think about this a bit like a gel forming in the cell, and this gel specifically labeled here is called paraspeckle. Paraspeckles consist of certain proteins and RNA molecules that bring them together, and they form without membrane, actually. We call them also membraneless organelles. And my lab actually found out that these physical interactions, the clustering, this gel-like formation in nuclei of cells that you can see here in these red labels, actually regulate stem cell differentiation. So I think this is, a very interesting example where physics and chemistry come together in order to regulate stem cell differentiation, not just gene diagrams. And in addition, this is also potentially very important for disease because the proteins that are within these paraspeckles are actually known to be affected in amyotrophic lateral sclerosis, ALS, and other neurodegenerative diseases. So this process indicates from basic biology, basic developmental biology, processes that may underlie actually neurodegeneration. I would like to give another example for a very basic question that I am very fond of, which is what regulates the speed of development. And I know it's a slightly busy slide, but I will walk you through. If you look here at the top, this is quite obvious. Our life cycle, humans and mice, we start as single cell embryos, and you know what comes in the end. And these phases, this life cycle, of course, starts with development. And what's very striking is that the time dimension in development is actually fundamentally different between different organisms. For example, to a certain, I would say, manifold of the embryo, it takes 7 weeks in the human, more or less, and 2 weeks in mice. And now, what's very striking is actually that when we take those stem cells, we produce these iPSCs from humans or from mice, this time delay or time, I would say, these time constants actually remain also in culture. When we differentiate the cells, for example, 40 weeks, more or less, 40 days, more or less, from the human, the developmental stage will be equivalent approximately to 10 days in the mouse. So this is, it indicates that there are intrinsic mechanisms that control developmental timing. And that's what we are after here. So just to tell you a bit about our approach, what we use a technique called single cell RNA sequencing in order to isolate single cells and to sequence all the genes that are expressed in that particular cells and their amplitude, their levels. And we repeat that. We have a process to do that by conjugating cells to these beads, as you can see them going through a microfluidic chamber, in order to characterize all the genes expressed by numerous different single cells coming from the culture. And this allows us to characterize what are the sequences very accurately, what are the sequences of genes that are expressed during differentiation process, for example, from different species, in order to create a map that explains the time delay or the time shift between organisms. Now, I think that's actually quite important in order to, because if we understand beyond this fundamental question, if we understand the beginning of life, we can also understand perhaps acceleration of aging. And of course, I'm pretty sure that most of you know the example of progeria, an accelerated process of aging. And I'm actually convinced that by doing this fundamental work, we'll find hints what is actually wrong with certain diseases. Now, on the application side, I would like to mention two interesting examples. I mentioned that we need to think about how to apply pluripotent stirrers in a safe fashion without immune rejection. So actually, already 20 years ago, I was actually the first to show that pluripotent cells, and you can just look at this red line compared to the control, they do express molecules that are important in rejection processes. It was actually then thought that pluripotent cells, because they're embryonic cells, they are not immunological, let's put it this way. But I've shown that they are immunological, and actually that immunology actually increases over time. So that actually exposed the problem of cell rejection in future therapies. So what we've done in my lab that is still active in Germany, we have applied for a project funded by the BMBF, led by me, called iPSCs, where we use mRNA technology, actually well before the vaccinations, in order to reprogram the cells from somatic cells to pluripotent stem cells. And the way we do it, we identified very rare individuals that have, I would say, low genetic complexity that makes them match to large proportion of the population. So this is because how the genetics works in the genes that determine rejection or matching. You can't do it for all the individuals, approximately only to 30 or 40% of the population. That's more feasible. And that's the approach that we are taking now. We have selected 3 individuals to start the donation process to make iPSCs under clinical standards. This is manufactured with regulatory agencies. They supervise the process in order to make sure that the cells that come out from this process are withstanding clinical standards. And we think that by creating these banks, we'll be able to match approximately 100 million people in Europe and from European descent. Another, I think, very exciting finding in the lab, we've made recently a breakthrough in making drug models more effective, of course, in a specific fashion. We found ways to create peripheral neurons that basically do that, right? Motor neurons and sensory neurons. So those are the cells that you can easily see now in the movie in front of you. You can see how homogeneous the culture becomes with the cells representing our peripheral nervous system. You can see how quick and homogeneous or uniform this process is. And I think it's actually quite amazing to see how these projections and how cells migrate and how they create these networks of cells very early on, again, representing our peripheral nerves. And I'm stating that they represent peripheral nerves because we have characterized them, again, using this approach, the single-cell RNA sequencing. You can see on the left here a map that shows thousands of single cells. Each point here represents one cell, and the whole genome that is being expressed in that single cell. And superimposed on that, you can see 2 genes that I'm only showing 2 genes out of thousands. Here on the left, SOX2, and on the right, I'm showing MAP2. So you can see, for example, MAP2 represents a marker gene expressed in peripheral neurons. So therefore, I've depicted here a laser beam saying that, look, from the undifferentiated state, those iPSCs, we've made a very homogeneous culture of peripheral nerves like a laser beam. So now the process is very efficient in making peripheral nerves. And this gives us an indication that we can use these cells now as a model for making effective drug development or toxicity, or to study, for example, diseases like amyotrophic lateral sclerosis. Now, I mentioned at the beginning that one very interesting aspect for me and many others, I hope, is the actual expansion of the iPSC impact beyond medicine. And here there is a a very significant problem, I think. We know that the rate of extinction, actually mammals, birds, animals in general, is actually increasing all the time. So that's what you see here in the science paper, the rate of extinction compared to the background extinction. And obviously, I mean, we all know that this is, I mean, mostly man-made extinction, either by poaching or environmental loss, and so on and so forth. So what is being done in my lab is the creation of a stem cell biobank. And the species that you see here are some of the species that we have in this biobank. And you can think about it as a bit like a Jurassic Park approach. It's a bit more scientific than the movie. We can't use DNA for this purpose, but because we have to have intact cells in order to make stem cells. And here shown on the top, an example where we have made from a northern white rhino induced pluripotent stem cells shown on the right. Just to inform you, northern white rhinos are functionally extinct. There are only 2 remaining specimens today in the world, both females that are infertile, which means that they will be extinct. So actually, relatively soon. So what we've done, we created these iPSCs as basically as a starting point to differentiate, hopefully, eventually, gonads and embryos in vitro. This is something that we have not accomplished yet, but so far we have very nice success of creating different cell types, for example, cells of the heart of the rhinoceros in the tube. And that's what you're seeing here in the center, cardiomyocytes that represent the northern white rhino, which is functionally extinct. So what we work on now is in networks of veterinarians and scientists trying to get these very rare species to start preserving them for research purposes and also for the extinction purposes, and also to work on the differentiation aspects and biological mechanisms in order to create embryos, which we think will be able to transplant in surrogate species, close species, for example, to another rhinoceros species, starting from the northern white rhino. And this brings me already to the end, and I said that to say that I think that iPSCs will take a center stage at drug research, and I wanted to mention that I think Leiden University is a fantastic place to do so, because there is here a strong effort to combine the LACDR with the LUMC in creating this new science in regenerative medicine center. And we work together very closely on accomplishing the topics that we discussed today in the Astellas building here on the campus. We are now creating basically the manufacturing sites and also the research sites in order to be able to make what I've shown you today a reality. But of course, we work with collaborations otherwise also in the LIC, in IBL, hopefully Lion as well, and so on and so forth. So my goal is to, again, to create a program. Maybe we'll call this LA Stem Cell DR in the future, that will go in stem cell therapy. We'll have dedicated courses in master and bachelor in stem cell biology. And again, to reiterate, the topics we think about are fundamental research to feed into applied research, to think about neurodegeneration and injury, immunological matching. I also mentioned diabetes, and the applied research will go here in the direction of automation, and of course, working with biotech companies. It's also a very important aspect, and economics, because we need to find ways to fund those activities, not just in university, but for society. With that, I'm coming to the end, and I have 2 questions that I'm presenting here for any of you who might be interested. I mean, I think this can perhaps steal some discussion or thinking. How would you think yourself to become involved in research and stem cell application, and how you would like to see the evolution and the involvement of stem cell science in Leiden University and in the Netherlands. And with that, I'm happy to take any question and discuss with you. Thank you.
Perfect. Thank you so much, Micha, for this. This was definitely very interesting, and very, very also promising in terms of what kind of implications can have in the near and maybe also long-term future.